details about this lecture. So we started this air quality management lecture series in the year 2020 October. First of all, I also welcome all the participants for this air quality management lecture series. And this lecture series provides an important forum for young researchers, scientists, engineers, and also academicians to share the latest advancement in theories, applications related to air quality management. So this lecture generally covers the recent developments, case studies, innovations, use of recent technologies in addressing the air quality management, and also provides an opportunity for the collaboration with various stakeholders. So this gives a glimpse of the past air quality management lecture speakers, eminent speakers from across the globe. This is the continuation of that. And yes, as we started this air quality management lecture series, then we also come to a group, and we started an air quality management association. This was inaugurated by our director, Professor V. Kamakoti, and also another eminent personality from Tamil Nadu Pollution Control Board chairman. So they inaugurated, and basically this air quality management association is to again provide networking, people who are interested to work in the area of air quality management. So various stakeholders involving industries, academicians, research scholars, and also the NGOs and other community developments. If you try to look at it, this air quality management lecture series started with a part of this clean environment for planetary health in Asia. This was a project we started in September 2019, which was funded by UKRE. And in that project, since it was a wider collaboration from various universities across Southeast Asia. And we made this to address various environmental problems by means of engagement, understand what works in a particular area, and how the partnership will help in addressing the environmental problems. So this was really provide a strong foundation for this air quality management lecture series. Okay, so since we also started working with this Indian International Conference on Air Quality Management, this year we are going to celebrate 9th anniversary. And this 9th Indian International Conference on Air Quality Management is scheduled on 18th to 20th December 2024. This time we are going to organize at IIT Madras. So I request all of you to block your calendar and watch out for the various activities planned in the conference. So now I welcome Harish. Now I request Gopika to introduce Dr. Gopika.
A very good afternoon to everyone present. It's my great pleasure to introduce the speaker for today, Dr. Harish Phuleria. Dr. Phuleria is an associate professor with 20 years of experience on environmental monitoring, exposure assessment, and environmental health at the Environmental Science and Engineering Department, IIT Bombay. His primary area of research is quantifying and characterizing the short and long-term exposures to different environmental stressors, such as air pollution and road traffic noise. His research group focuses on understanding the emissions from vehicular and biomass emission sources, monitoring and modeling the long-term air pollution exposures in various microenvironments, quantifying toxic chemicals in human tissues, and examining the associations of air pollution exposures with health effects in children and adults. His specific interest lies in characterizing new exposures like that to ultrafine particles, to brown and black carbon, and to chemical constituents in aerosols of different source origins, in particular to traffic exhausts in urban areas. The talk for today is titled Exposure to Urban Air Pollution in India and Children's Lung, the APEAL study, where he'll be presenting the association of lung function and air pollution exposure to urban adolescents through the air pollution exposure on adolescent lungs study, or the APEAL study. It's a longitudinal multi-centric cohort study conceived and being conducted in 4 major cities of India. Sir, we welcome you once again. Over to you.
Okay, thank you very much, Gopita, for the nice and long introduction. Actually, let me first share my screen again. I hope this will work. Okay, so you were able to see my screen now?
Yes, sir.
Yes. Yeah.
The presentation.
Are we recording?
And I'll just swap. Now you should be able to see the full screen, yes? Are you able to see the full screen?
It's in presenter mode, sir. You can.
Okay, let me see. What about now?
Yes, sir. Good.
Okay, very good. So, good afternoon. A very warm welcome to this lecture series, which I'm very happy to be a part of, and really want to commend Professor Shiva for really holding this for so many sessions in last several years. And what I'm going to share today with you is some of our current work, which is very close to my heart, which is to kind of take a dig on how air pollution affects our health. And in this particular case, we have a study which was funded a few years back by Wellcome Trust India Alliance, where the interest is to examine the effect of air pollution on children's lungs. Where our interest is to not only examine the effect of air pollution, but also some other kind of factors like physical inactivity, nutrition, and some of the others. I am going to focus mostly on air pollution. This is basically to share with you like the study design, the questions, and the motivation, etc., and a few preliminary results. I mean, it's a very ambitious effort. For example, we are planning. We have already completed the first, recruitment of subjects and assessment, which is about 4,000 subjects so far from 4 study areas. And like slowly, like for example, the data analysis is undergoing. So I will share some of that. Now, this is a picture which is from Mumbai. This is like very close to where we live, where our IIT Bombay is. And I'm sure that all of you who are part of urban areas will know this kind of a structure, right? So basically we have high-rise buildings along with, low socioeconomic housing, that the extent could be different. Sometimes we may have greeneries. You can see this picture is taken from our place. So you can see some of the trees also there. So it's like different land uses there, but most of the cities, for example, I was reading a recent report from Bangalore, where, for example, from 1975, when the total vegetative cover was about 70%, in 2023, it has reduced to about 2 to 3%, right? So obviously the city landscape is changing. And that is also, which also kind of raises concern, because that means economic growth, that means new sources of emissions and air pollution levels, right? So I am teaching at IIT Bombay in Environmental Science and Engineering Department. And today I'm going to share about how how exposure to air pollution is being conducted in this study, which is called the APEAL study. I will tell about the study later. First of all, I would like to acknowledge, so this is a multi-centric, multi-investigator study, which is being conducted in 4 cities, Delhi, Mumbai, Bangalore, and Mysore. Professor Mahesh, who many of you may know, is the lead principal investigator. Along with him, Professor Rajesh, both of them from JSS Mysore, Professor George from St. John's Medical College, and Professor Anand from AIIMS New Delhi. And then from IIT Bombay, I am participating in that. I would also like to acknowledge, so this is like a work which we are doing together. So obviously like a great leadership is provided by Professor Mahesh, and I think huge support from all the study centers. I would like to also acknowledge that my team at IIT Bombay, you can see the picture, and also all other teams, for example, at JSS Mysore, at AIMS New Delhi, and St. John's Bangalore. I think we have about more than 50 total people who are supporting this study. Obviously, I would like to thank the study participants, parents, without which this study could not be possible, and also DBT Welcome Trust India Alliance for really supporting us well so that we can conduct the study. A snapshot of what the study is. So the APEAL basically stands for, as you can see in the black bold, air pollution exposure on adults and lungs. So this is a longitudinal effects of air pollution exposure on adults and lungs study. So it's a cohort study, as you can see here. We are investigating the lung function of children which are recruited at the age of 10 to 13 and then following them for next 4 years. Initially it was 5 years, but because of COVID and all of that, I mean, we were really delayed. So this is now reduced to 4 years. Idea is that their lungs should grow naturally. As we know, the lung growth happens. But then, for example, those who are like, let's say, very highly exposed areas like Delhi, compared to, for example, Mysore, should have a relatively lower lung growth. That is the primary hypothesis of this study. You can see the investigators, the institutions which are shown on the left, and the study centers. And in terms of air pollution, our major focus is on, as it is an urban area, so we are focusing on traffic related air pollutants. One of the major ones that comes to your mind is nitrogen dioxide, and the another one is fine particulate mass concentration, PM 2.5. So we are monitoring the lung function. We are basically measuring the biochemical markers in blood. Then to relate that with the lung function, we are collecting data on lifestyle factors like diet, exercise, which can influence the lung function in addition to the exposure to air pollution, right? A little bit of a background. So it is pretty well now known that Indians, for example, like if we compare them against the Caucasians, like in the Europe and Northern America, so we have about a quarter lower lung function. So our lungs are lesser developed in a sense, you can say, or the capacity of the lungs is lower, right? So that is natural, that is a genetic difference. And the lung function are closely related to all-cause mortality and development of something called chronic obstructive pulmonary disease. So why we are focusing on lung function is because this is like an early indicator. Those, for example, who have lower lung function, more likely they will have a lesser longevity. For example, they may prematurely die compared to those who have better lung functions, right? So that is what has been kind of known, is already known. What you can see in this figure is, for example, how the lung function trajectory looks like. So as you can see, the lung basically grows in the first few years to up to adolescent, let's say up to age of 19, 20, and where it reaches the maximum growth and the maximum function, lung function, the peak expiratory flow rate, etc., are achieved there. And then as we age, the lung function declines. Okay. Now, this decline in the lung function can be affected by multiple factors, including air pollution. And the idea is and there are a number of studies actually which have examined the adults, which is the more than 20 years of age, where we are continuously focusing on the attenuation in lung function decline. While our interest was in the first part on the left, which is the dotted area, where how the lung growth happens, and those, for example, who may have some risk factors like exposure to high levels of air pollutants, or maybe physically inactive lifestyle, or for example, dietary inadequacies, or for example, exposure to secondary tobacco smoke, etc., they may have a lower growth. Okay, so that is what we want to capture. This is particularly kind of telling about like how this trajectory looks like. And there are a number of factors like genetic and host factors I already mentioned. For example, diet, physical activity, whether they have pre-existing conditions. And then also like, for example, even the early life factors, like if those who have preterm births, or for example, those who have, let's say, lesser weight at the birth, right? early life environmental exposures. If their parents were smokers or working in occupational environment which have high exposures, right? So some of those can also affect this lung growth trajectory. Now, this is like what is one of the motivations for this study. This is from Southern California. You can see here the NO2 concentration on the x-axis and the growth in the lung, which is measured in terms of the forced expiratory volume in 1 second. Okay, in 1 second, how many milliliters of air is, let's say, exhaled. So that's what the y-axis tells. And what it shows that as the NO2 levels increase, there is a decline in the FEV1. So that means those who are living in areas with higher level of NO2, so they have, for example, lower lung function, right? It was further, for example, seen, let's say, because, I mean, as we can argue that this can depend on a number of factors. For example, one of the major factors is height. Because those who are taller, they will always have higher lung function. That is one of the reasons why the Europeans and North Americans have always higher lung function on average compared to the Indians, because our height is shorter. So what we do, we look for the predicted values of FEV1, which is adjusted for gender, age, and height. Okay. And what it shows on the y-axis is the percent of the people who, in this case, children, who have the FEV1 less than 80% of the predicted value for that age, for that gender, and for that height. And what do you see that on x-axis is PM 2.5 concentration? It is now other way around. It shows that as the concentration of PM 2.5 increases, or let's say the exposure to PM 2.5 increase in these different neighborhoods, you can see that there are more and more people who have compromised lungs. That means their FEV1 percent predicted less than 80% is more, right? So this is like what is really one of the seminal studies which was done back, I think, late 90s and early 2000, which was published in one of the, I think, highly reputed journals, New England Journal of Medicine. But one of the problems, if we want to really kind of extrapolate this study to where we live, or in most of the countries and cities in rapidly industrializing countries like India, China, or for example, even low socioeconomic countries. You can see that here they are stopping at 30 microgram per meter cube. Now, how many of you have actually been living in areas in India or cities where you have average annual PM 2.5 is less than 30 microgram per meter cube? So we don't know what is happening beyond that. And that's one of the problems that we wanted to take up, that what happens after that in our cities where, for example, the levels could be sometimes 3 to 4 to 10 times higher than where the investigations have been done. So if I summarize all of this, a number of studies are there which have identified association with low lung function with air pollution. For example, countries like Netherlands and some of the Balkan countries, there they have found that 50% of their air quality is like basically less than 50% of the EU limits, but still there we are seeing the effect of air pollution on lungs. Our expectation is China and India are relatively highly polluted, so we expect more severe adverse effects. But is that true? Now, the lower pollution levels seems to cause higher adverse effects in some population, while others exposed to higher pollution levels are less affected. This is also seen. So there is not a consistent kind of relationship even in the adults. So this raises the possibility of maybe modifying factors that may mitigate or worsen the effects of air pollution on lung function. I will just give you an analogy just to keep in mind, even though it is not directly related to that. But for example, you think about traffic noise. And let's say if you want to know whether people are affected by the traffic noise, whether there's stress levels, blood pressure, heart rate variability, cardiovascular outcomes, which is like there is a huge literature on that, is adversely related, right? These are. But what is found is like those cities or areas where the noise levels are lower, people are more sensitive. So there is adaptation also, which is there, right? So for example, how, let's say somebody who is living very close to a a major road will not open his or her windows. Now that will have effect on the exposure to the traffic noise inside the home. The same thing could be also for air pollution, right? So in that sense, there could be other modifying factors. That is one I said, but then it could be others also. But we should always remember that there is no safe lower limit of air pollution exposure. I'm sure that some of you have come across. So yesterday only, US EPA has revised their fine particulate mass concentration standard, annual average PM 2.5 standard for US to now 9 microgram per meter cube. Okay, it was 12 microgram per meter cube earlier. It is reduced to 9. What does it say? It says that if the values or the level of PM 2.5 are more than 9 microgram per meter cube, it is not good. It is really adversely affecting, right? Even though we should always remember there is no safe limit. So what is our primary hypothesis? It is exposure to high air pollution, for example, exposure to fine particulate mass concentration, impair the lung development, leading to significant deficit in maximal attained lung function in adolescents. So that means by the time they reach their peak lung function, let's say about the age of 17, 18 or so, that is the age where we will capture them in our study. So those who are living in higher air pollution area, they should have impaired lung function. That means their lung will be lesser grown compared to the others. And a portion of the population may be highly susceptible, while some may be highly resistant to the effect of air pollution. And that is exactly what we are also finding. And I will show you some preliminary studies. I mean, it's too early to quote them, but I think these are very interesting findings. So what are the research objectives? The first one is to conduct a prospective cohort study, which means like you take a number of people, then you measure them at time point T1, then you follow them for time point T2, T3, T4. In our case, it is year 0, that is the baseline, then first year, second year, and third year, right? And we are looking at the growth in the lung function. It is 11 to 13 years, which will be then becoming from 14 to 18, and over a period of 4 years. And we have selected these 4 cities, understanding that Mysore kind of represent moderate air pollution, Mumbai and Bangalore high, and Delhi very high air pollution, right? As far as the PM 2.5 levels are concerned. Then the second objective is to assess the long-term exposures of traffic-related air pollutant. What is the meaning of long-term? That means, for example, annual or relatively like not as like a peak or not like for a day or a few hours. Something which we can say typically a person is exposed over the year, right? So that assessing that. And the characteristics of the PM 2.5 like chemical composition. For example, some of you may have also come across oxidative potential, which is relatively like probably in the last decade has been really coming up as a new surrogate for exposure to air pollution and the respective health effects. And this is what we are going to do in schools and homes and their correlation with lung function growth in the children. We have other objectives like measuring blood biomarker, for example, inflammatory biomarkers so that these could be used as in the clinics also as a biomarker to predict lung injury later in life. And also to evaluate other individual and community level factors, as I was mentioning, socioeconomic status, home characteristics, physical inactivity, pre-existing conditions, and also nutritional inadequacies. But today I'm going to share mostly about the study design and the exposure assessment part. Now, what was done? We recruited, for example, about 5,000 subjects. Screening was done for more than 5,000. Then some of them did not give consent. So then the eligible subjects were there. Then we asked them different questionnaires to get the data. I will show you. A spirometry was done about, this is a little older data. I mean, this is now almost like 3,800. So about 3,000 subjects where the spirometry, that means the lung function assessment using an objective portable spirometer. We collected biosamples like blood, hair, and nails. About 3,000 subjects we did that. And you can see like why, for example, the subject size reduced to about 3,000 because many participants were not able to perform the spirometry, some withdraw after some of the tests, etc. Some of them did not agree to give their hair and nail samples, etc., right? All of those, or blood samples as well. You can see we have a battery of questionnaires. These are different components from demographic information, socioeconomic status, spirometry, history in terms of allergies, medication, vaccination, anthropometry, for example, weight, height, the body fat composition, general clinical examination by medical officers, nutritional status assessment based on food frequency questionnaires and dietary recall. Then examination, this is again clinical upper respiratory tract. Then you can see some other examination, again clinical. And then more questions about the potential, things which can affect the exposure. Let's say child's bedroom, where, for example, with the orientation of that, whether they have some pets, etc., which can induce allergies, or for example, whether they are living near a road which has, let's say, trucks or major vehicles passing, then dietary pattern, all of those, physical activity, air pollution questionnaire. You can see like number of total 217 questions were there, which is pretty actually challenging for both the participants as well as those who were administering them. So in general, it took about 2 days.
Disconnected, huh? We just wait. Harish, we lost you. It's, okay. Thank you. Right. So there is a power failure there. Inform them. Few minutes. There was a power disruption at IIT Bombay. He'll join in a few minutes. Join, huh? Hello, Harish. Can you hear us?
Are you able to hear me back? Sorry for the interruption.
Yes,
Yeah.
we can hear.
Okay, great. So let me just share. Okay. Are you able to see the full screen?
Yes.
Yes?
Yeah, yes, Harish, we can see you.
Or this is the full screen?
No, sir, the first one.
Okay, okay, sorry. Somehow it's not showing. Okay, good. So we were here. So I mentioned about like the tools that we are using. So the lung function is being assessed in children, as you can see through the spirometer here with the trained technicians. We first measure their anthropometry, that means like height, body weight, etc. Also, what we do is first we measure, we call it pre-bronchodilation. That means we measure directly. And then after that, there is some kind of a drug is given, which kind of opens up their airways, and then we measure it again. So that kind of ensures the reversibility of, let's say, any kind of obstruction in the lungs. Each child is given 8 trials. If they are able to do that, then good. If not, then either we drop the children or the child, or for example, we do it at the next session, because this is an effort-driven maneuver. So it is like they have to really blow, and there's a way it can be done. It is not as straightforward. Of those who have any kind of surgeries done or having cough, cold, etc., are excluded from the study. Just to kind of give you a feeling of it, for example, this is the data, which is, I mean, preliminary, again, I want to emphasize that, which is showing the gender stratified lung function levels in 4 cities, as you can see here, Mumbai, Delhi, Mysore, and Bangalore. And you can see that all the gender differences, you can see there's a star, which is showing that there is significantly statistical difference between the lung function of the male and female, which is known because the anatomically, for the same age group, males have higher lung function compared to the females. So that is what we have seen. And also we see that, interestingly, you can see that if you see across, let's say FEC, the first row here, you see that Delhi has the highest lung function, 2.53 liters. So now that is quite high, and you see compared to that Mysore 2.17. What is our conventional wisdom says, and where we started this study? We thought that it should be the other way around. So the Mysore people should have the highest lung function because they have the lowest air pollution exposures. While Delhi has the highest air pollution exposures, they should have the lowest lung function. So this is a kind of a very contradictory or counterintuitive finding. And this is also, I mentioned earlier, that there could be a number of other risk factors and also adaptation, which is maybe responsible for that. Because our understanding of how air pollution is affecting health is mostly driven by the studies which are in Europe or North America, where the levels are really, really low, right? So extrapolating them to our levels and saying that the burden is this much is, we should be very cautious about that. So, I mean, one of the takeaways I want to give to at least those who are relatively new in this field is whenever you look at the numbers from whether they are published in Lancet or coming from GBD, coming from WHO, like you should take the results a bit with the pinch of salt because, for example, there are almost like very few, very scarce literature from our study areas, which really supplements the findings which have been kind of used in those assessment. Of course, they try to extrapolate it from biomass smoke studies inside homes in rural areas or smoke through the cigarette smoke, etc., etc. But that is not necessarily, it is not those response is not very well established. Okay, so this is very, very important, just to keep in mind. So as I was telling you, it can be affected by age and gender. So what we looked at the percent predicted, which is adjusting for the age, gender, and height. And again, we classified them into 3 groups. Those who are having normal mean predicted, that means like up to 80%. Those who are having less than 80%, which is, that means compromised lung function. And those who are having actually more than 100%, that is supranormal. That means their lungs are even better, right, than compared to an average for that age group, for that gender, and for that height. Now, what do you see? If you look at the FVC, the 3 left columns, you see that Delhi has the highest number of supranormal kids. That means those who have really very, very good lung function. even better than the average, right, of the Indian population. And you can also see that they have the lowest number, I mean, almost similar to Bangalore, number whose lungs are compromised. That means the lung function is relatively better. And if you see, for example, Mumbai shows and Mysore, both of these show relatively very high proportion of kids who have compromised lung function. Similar is the case we see for FEV1 and also the mid-expiratory flows, which is the FEF 2575. So 50% of our subjects in 4 study areas show normal lung function, and Delhi and Bangalore have the maximum number of subjects with this very high supra, like better lung function, which is not known, and we don't know why that is happening. We tried to run a mixed effect linear model, and it is very interesting. You see that this is the coefficient for the effect of air pollution on lung function. So you can see that is a positive coefficient. What does it mean? Higher the air pollution, higher is the lung function. That is total counterintuitive, right? That is not what we saw from the studies there. And that is because it is driven by the higher lung function in Delhi. So one of the things we found is that how the average height of the children in Delhi is higher. And at the same time, there are differences, for example, other than that, which could be in terms of nutrition, physical inactivity, etc. But we are not quite sure. Socioeconomic also, because even though we tried to use the same methodology for recruitment of the subjects, because of the COVID, we then slightly deviated from that. For example, I will show you later how we did that. And in that sense, we see that the significant association is there, but it is totally opposite. It shows positive effect. So and these are based on some of the pilot measurement. These are not actually the individual exposure of each of the children, which is the next thing which we are doing. We are making models to get the exposure of everyone so that we can actually look at the true association, right? And these are adjusted for some of the factors, but not all of them. And so they may also play an important role. If we just focus on, let's say, Delhi and Mumbai, you can see that this right plot here, it is showing the winter time concentration of Delhi and Mumbai. Delhi is significantly higher, as you can see here, and a very large variation, right? And it is statistically significant, highly statistically significant. But if you look at like the quartile distribution of the lung function, it is pretty much similar and shows a little slightly positive trend. So as we move from the lowest quartile to the highest quartile to the PM 2.5, the percent predicted FVC is higher. So that is again not like something that we were expecting. We are not seeing any decline in the lung function with increasing levels of PM 2.5, and there could be a plateauing effect. So for example, after a certain high levels of exposure, it is possible that there is no further impact of air pollution on lung function, right? And this is the same story is for the FEV1. This was for FVC. We tried to look at the relationship between body mass index. So for example, as I said, the height, weight, and some of those also kind of make an impact. You can see there's a positive relationship, and the underweight children exhibit supranormal lung function. So those who are actually underweight, I mean, we understand that they don't have their growth trajectory is actually compromised, but they are showing supranormal lung function. That means they have better lung function than that. So there is some kind of offset which is happening. It is still under investigation. We don't know the reason. If we try to see that in perspective with the studies other places, they don't have underweight children. Okay, so they have mostly normal, overweight, obese, and or super overweight and obese. So one of the things we are also kind of hypothesizing right now is that our obese classification or overweight children could be quite different from what is there in the Caucasian population, where some of these results are there. So that was like just a snapshot of, for example, what we have done in terms of this study, in terms of collecting data from multiple centers for lung function from children. So right now we have completed about 4,000 subjects for the baseline, and the follow-up is happening. About 25% of the subjects have been done for the first year follow-up, so that by the end of this year, we'll have the second year follow-up done, and we will be able to look at the change in the lung function. So let me just show you a few slides about exposure assessment. This is the uh, overall plan, how we would be doing that. So for example, identification of the study areas, which I already mentioned to you, and identification of sub-study areas. For example, when we are saying Delhi, Mumbai, Bangalore, Mysuru, we are not looking at the full Mumbai, full Bangalore, Delhi, etc. So what we did, we said that we followed the BOLD protocol and said that we will take up a population which is having at least 2 lakh population. Now, Mumbai, Delhi, Bangalore, all these have really, right, in crores, the population. So what we did, we went into the ward level and then selected a few representative wards in some places. In case of Mumbai, we looked at the S ward, which has a population of over 7 lakhs. Then we had a strategy to randomize, for example, the subject selection, which is based on, for example, neighborhood identification, road network, etc. And then once these were identified, we went for the consent. So we went for the consent from the children. Those who agreed to participate, consent from parents and assent from the children. That means both of them need to agree. And then after we have this set of actually the target population was 1200 from each of them, so 4800. So we have about, when we had about I think 800 or something, we started for the PM measurements, and we took for the consent for PM, and we said that we will look for a subset of homes, for example 60 of them, which is again we will select semi-randomly, kind of capturing the heterogeneity of these locations so that we can get the exposure differences. And we aim to measure, we have done that bi-weekly 14 days or 7 days NO2, and PM measurements were done for 1 week during the pilot, where we saw that the 24-hour, typical 24-hour exposure is giving pretty much close to the weekly. So we then left with the 1 day exposure on each home. This was done for the 2 seasons, summer and winter. And similarly, we collected hair and nails. The idea was to really look at some toxic heavy metals in them and then relate that with the heavy metals in the PM to just understand whether there is a relationship. Chemical analysis of the collected PM and hair nails, and oxidative potential as a toxicity measure. Looking at using the GIS, road network, building and population density, the built environment, etc., to kind of generate information which can be used along with the 60 locations to make a statistical, or what we call stochastic, land use regression model. So point is that you have, let's say, locations which are here. This is an example of another study. So if we have 60 locations and you have, let's say, these 60 locations are part of about 1,000 or 1,200 children. So from those 60 measurements and the other kind of surrogate measures which are easily available through GIS, etc., we can make models which can really enable us to get the exposure of everyone. And simple tools, I'm sure many of you are familiar. For example, low volume air samplers like Minivol, having about 5 to 6 liters per minute of flow rate. Mostly we used PTFE Teflon filters, but sometimes also quartz filters for ECOC analysis. We use some low cost sensors for indoor measurements, and they give real-time measurement. These were all first and 24 hours. And you can see here, this is the small tube here, which you may be able to see, which is a passive tube where the gas kind of diffuses inside and kind of allows us later in the lab, we can do Saltzman method estimation of NO2, right? So with this, like, we really went ahead, and that was what we were able to do. So this was the work plan. So we really did 2 cities at the same time because the limitation of the equipments, etc. It was not possible to do everything. So we did 2 cities at a time. We did winter measurements in November 22, February 23, because we were not able to do it in 2 of the cities in the first winter season. So we just did just now in February 2024, and the summer was done in March to June 2023. About 60 homes in each city were measured. We did gravimetric measurement, real time using the low cost sensors. These were done outdoor as well as indoor, some of those. And NO2 was done for 7 to 14 days, both indoor and outdoors. This is a simple example of like how, for example, this is the S ward in Mumbai. This is the kind of central South Delhi. This is the Bangalore, I think one of the BTM around layout there. And then this is Mysore. Mysore was taken full. And the dots are showing, for example, the yellow are the subject location, the overall subjects where the assessment was done for health data, health measurements, anthropometry, etc., etc. And the red dots are where the exposure measurements were done. And the idea was after these exposure measurements, we should be able to make models to get the exposures of these. This is a further like detail. Let's say in Mumbai, for example, we selected this S ward here. And this in the S ward, we have several sub ward in information. For example, almost 14 sub wards were there, which we selected all of them. And then what we did, we said that we will recruit population representative numbers. So this was 7 lakhs, and each of the sub ward we know the respective population. So if we wanted to select, let's say 1200 subjects, we said that those which have 2 lakh population, it will be 2 lakh divided by 12, 7 lakh multiplied by 1200, right? So the proportional number of subjects were taken from different sub wards, and then we went ahead with the kind of consent and measurements. As I was telling you, we did multiple things. What is not sure, yeah, it is here. So because the idea was initially that we will do school recruitment, which is easiest. We first enroll and get permission from the school, and then basically conduct the camps in the school, do all the assessment. But then what happened was it was actually the money was received in December 2020, and one, it was anyway lockdown periods, and people were already pretty apprehensive about other people. For example, some of the our team members were from different parts of the country, right? So they were going to Mysore and Bangalore homes speaking Hindi and English. So obviously we took help from the local people, but it was pretty like the response was very low. So, and then the schools were closed. There was no information from the school. So we thought, okay, what should we do? So we went door to door. And here we really got support. I mean, it is not political, actually, totally apolitical. We took help from the shakhas of RSS or Shiv Sena. And because they have these, presence in multiple neighborhoods, we kind of explained them this study, and they actually helped us to kind of find us a place where we can organize these camps. But also we did it inside the homes of these people. For those who are not familiar, many homes, actually almost 70 to 80% of our children in Mumbai are coming from slums, where they have only one room, maybe 4 to 5 people in that. There is very less space. So I think this is like a commendable kind of effort by the field teams. We went through NGOs, other like social workers, etc., and we tried to kind of collect the data from those people. So you can see like how the people from our team went to different places, talked to children. Sometimes they were in open camps, sometimes inside some schools, sometimes in homes, shakas, etc. The hair and nails were collected, then its standard extraction, and then ICP-MS was done. One of the important thing which we did was we said that from the very beginning we want to have the data totally digitized. We don't want questionnaire which are on paper. So we used some kind of like survey apps, like Go Survey is what we are using right now, where we built the questionnaire. It was validated multiple times, and now every data which is collected is actually going to to central server, and from there we can actually kind of get it. And we have barcodes for each of the children. So one identity for everything, whether this is a blood sample, whether this is a hair and nail sample, whether this is a air pollution measurement, or whether this is a, for example, clinical examination. And we gave them some kind of a health card so that the children feel like, okay, they are getting something. And we had a master sheet to track the progress. And some kind of a dashboard to know where we are. Like we did some pilot measurements. I already mentioned about these things. There, one thing we also did, we used some passive VTX tubes, which are basically charcoal impregnated tubes where we can absorb, let's say specific, VOCs, but specifically through the GCMS, we focused on benzene, toluene, ethylene, and xylene. Let's say these are the 4 areas. Then we went there, we digitized the road network. I mean, it was available, I mean, as geocoded. Then we took the consent, find the suitable homes. We went there. I think I showed you already that. So this was Mumbai, Delhi, Mysore, and Bangalore, right? So measurements were done. First, we did non-destructive analysis like mass concentration, as you know, pre and post weight. And if you know the flow rate and the time sampled, you can get the mass concentration. Black carbon assessed through this instrument called OT21 optical transmissometer. It basically measures the transmission of the light through a filter, and more is the absorption. So basically it measures in 2 wavelengths, in IR region for black carbon, in the UV region for what we call brown carbon, or brown carbon plus black carbon, or what we also call pH adsorbed, kind of substances. And we also analyze the water soluble organic carbon by extracting them. We analyze heavy metals using ICP-MS, and ECOC using ECOC analyzer. So many of these methods were used for characterization. This is like how you can see the samplers were deployed at different places. These are the regional sites at different institutions, because we wanted to ensure that we have these sites where we have no residential impact. So these kind of help us to understand the temporal effect, if at all, from some of those questionnaires I already mentioned. But let's say in terms of which were related to air pollution, we asked about which floor you live, number of windows, doors, etc., whether kitchen you have separate or same where you live, how many times you cook, how many hours, ventilation, and whether they know that they are affected by air pollution, etc., etc. So those kind of questions were asked to those people. Don't really go by these numbers. This is just a snapshot of what we are able to see. First thing is, like, the air pollution levels are same as how the study was conceived. The study was conceived, if you remember, I said that Delhi, very high levels, Bangalore and Mumbai, high, and Mysore, moderate, right? And we see that Delhi has the very high levels here. I mean, relatively, you can see here, this was done in summer as a pilot, and Mysore has the lowest, and Bangalore and Mumbai are similar. These are residential outdoor, okay? And what we used earlier was central air quality monitoring station data from CPCB. So you can see that they really follow. But when we compared those, we found that the residential outdoor locations have higher levels than the central site, which all of you who are from the field, you know that what we are exposed at our homes or schools or workplaces may not be represented by a central monitoring station, right? So there is a requirement for doing a very good exposure assessment. If we look at, for example, the sites characterized by traffic, urban background, regional, so you can see there's like the regional site, which were the actual, this was IIT Bombay, this was St. John's Hospital, this was JSS Mysore Hospital, and this was AIMS. You can see they are always lower. So essentially the residential areas have higher levels compared to where you do not have any like known source, right? So that was what was seen. If we look at black carbon, shows kind of pretty much similar finding what we have, and again regionals have lowest. The traffic sites have relatively larger, but it was not consistent. For example, Delhi, and it was pretty similar to urban background. Mumbai, it was higher, but in other places was not so. And one of the let's say explanations we have is probably these classification of traffic and urban background are not necessarily accurate. So we have been revising that. This is for, so this is outdoor black carbon. This is brown carbon, as I was saying, this is in the UV range, pretty much similar. I mean, the trend is very much same, right? So that is what one thing we can see. We measured the oxidative potential, for example, using, this is showing the DTT assay. It's, yes. Yes.
Good.
Is there a question?
No, please go ahead.
Oh, okay. Yeah, I think probably I will be able to finish in next 10 to 15 minutes. Yeah. So this is basically showing again from the pilot data. You can see that how the oxidative potential, which is actually a measure of the constituents in PM which have a toxic nature, which are redox active. So the whole hypothesis is that when the particles are inhaled in the lungs, then there, for example, they are responsible. So there is a disbalance in the reactive oxygen species because the oxidative, which causes the oxidative stress, and that leads to inflammatory kind of responses, which leads to certain kind of ailments. So, and this is a very relatively, you can say, easy to conduct in vitro, acellular in vitro test, which can be done in very relatively, I think, easily. And we see that now compared to the PM levels, you do not see the same thing. So for example, if you just look at the volume normalized here, you can see Delhi and Mumbai has very similar toxicity. I mean, right now, just call this as a toxicity. And you can see Bangalore and Mysore have similar. While we saw that the Mysore has the lowest levels and Delhi has the highest. What does it tell? It tells that the total mass concentration, which is assessed by the fine particle mass concentration, is not necessarily, all toxic. It may have also some, you can say, non-toxic or substances which do not necessarily affect. For example, salt in Mumbai, but still we see like some of the others or some dust components which are not so redox active like that, right? So similarly, we saw for the sites, etc. And obviously this is pilot, so this gives us some kind of information, but still, I mean, more investigation is required. We look at the heavy metals, etc. Overall, Delhi has the highest levels. I think there's a problem in this unit. I don't think that is correct. This should be in nanogram per meter cube, I think. And you can see like zinc is pretty much higher every place. You can see some of the manganese is also seen in some of the places, and maybe these are coming from higher amount of traffic in those places. But again, this is only water soluble from the PM 2.5. We don't know the total, and so it just gives us some impression of the differences. When you look at the NO2, we looked at both indoor and outdoor. Again, you see Delhi has the highest levels, Mysore lowest, and Bangalore and Mumbai similar. But other thing you notice is that the indoor levels in all places are higher, almost like 50 to 60% more. So what it is telling that if so your central air quality station is measuring the outdoor air and also at a one fixed place. So if you say that people in Bangalore or Chennai or Mysore or Mumbai or Delhi are exposed to those levels, that is incorrect, because they may be exposed to higher levels because of the sources. And I am sure that you can guess what is contributing to the NO2 inside the homes, right? It is coming from purely combustion. So there must be some combustion source, which is mostly gas stove cooking. So we saw, for example, for different type of sites, etc., also something similar. We look at VOCs. Again, Delhi showed higher concentration. We did not have enough number of samplers. We only looked at 3 cities, Mysore, Bangalore, and Delhi. And I would say that a similar trend, which are relating to traffic as one of the major sources, is kind of coming from all of these. Right? We look at these low cost sensors, indoor levels, and this is showing the PM1, PM2.5, PM10. This is Atmos, relatively, I will not call them low cost in the strict sense because this is almost like 40,000 rupees. But yes, compared to, for example, a BAM or an instrument which is causing like 20 lakhs or something, this is just cheaper. What we see is like, if you look at the 4 cities, again, Delhi has the highest indoor levels of PM 2.5. Bangalore and Mysore and Mumbai are similar, Mysore lower. So it follows, right? What was seen outdoor by a different instrument, which was a reference method, gravimetric, but the indoor level through real-time, low-cost scattering-based instrument shows the similar. Obviously, always whenever you are looking at low-cost sensor data, you should always be careful about the absolute values because they may be off, right, from the actual. We looked at like how the variation is happening. Sometimes you look at if the sources are really causing some of them. So sometimes we are able to identify some of these. This is still, I think, a lot of work in progress, but we are able to match some of the events in the homes to the peak events which we are able to see. This is like very interesting graph which I want to show you as just, so this is like here the dot size is showing you the PM 2.5 concentrations. And you can see that this is going from less than 60, which is, that the daily PM 2.5 standard is 60 microgram per meter cube. So if something is this small dot, that means it's fulfilling the daily criteria. But if it is larger, then for example, if it is more than 300, like some of these big size, it is like almost 5 times more. Compare that, for example, in Delhi, almost all sites are 3 to 4 times higher, right? I mean, 4 to 5 times higher. And there is a very less heterogeneity. You can see in Mumbai, you have some of them are higher, some of them lower, like that. And if you look at the NO2, very, very similar. Again, you have higher levels of NO2 in Delhi, but the variation is less. So which is telling that people are exposed to similar levels. So you can say that if the outdoor levels are pretty much comparable to this, then your central site is pretty much representative of them, right? But if it is showing heterogeneity, it is not. So that will have implications, because then you cannot use fixed site data for assessing the exposure and looking into the health effects. We look at chemical composition. I think you are aware about that the bulk composition comprise of the carbonaceous matter like elemental carbon, organic carbon, ions like nitrate, sulfate, etc., and elements. And this is showing you the distribution. One of the key features, what we saw, is if you look at Delhi and Bangalore. So for example, this is Bangalore and this is Delhi. Even though the levels you can see, this is 3 times higher here, right? Sorry. This is Mysore. This is Bangalore. So if you see that this is almost like 80% higher here in Delhi, but you see the EC and OC are pretty much comparable between Bangalore and Delhi. So if these are the ones which are really responsible for some of the effects, then the people shouldn't do those two places should have actually the same health effects, regardless of the actual PM 2.5 concentration those places. I think this is a pretty much kind of well-established theory and understanding on this topic. The last slide I want to show here is about this toxicity assessment. I mean, I showed you some earlier, but this is showing you how, if you compare that against these dots, these red dots are the PM 2.5 concentration. You remember Delhi has the highest, about 100, and Mysore had the lowest, about 35 or something. But if you look at the toxicity by two assays, which is ascorbic acid, DDT, and then a new cellular assay, which is done at JSS Mysore, they show very different responses. For example, if you look at ascorbic acid, Mumbai has very high toxicity, while Delhi has almost similar to as Bangalore or Mysore, right? So it is kind of raising questions about the suitability of PM 2.5 mass concentration as a good preventive surrogate of exposure to air pollution. We can develop predictors, as I was telling, because we need to make models. For example, this is showing you that how the satellite data can be retrieved. Like we can use the normative difference vegetation like NDVI to really classify the built environment and use that as a feature to make some of the models. This is one of the predictors, right? We looked at the hair and nails, heavy metals and PM, etc. I mean, some of those were done. So I will just summarize like the effort right now. I mean, I just want to kind of give you a sense of what was accomplished. Still, I think a lot of this will hopefully potentially lead to very good scientific publications and potential information which can be utilized for policy purposes, etc. But in terms of the effort, we conducted pilot measurements in all the cities, about 30 homes, and comprehensive characterization and oxidative potential was assessed. Almost like 41 species were assessed there, and now this has been extended to almost 120 homes. Oxidative potential was assessed in all of these places, and we looked at the relationship among them. Winter and summer measurements were conducted in about 240 total homes in 4 areas in 2 different seasons. We also measure indoor PM using the real time. So you have 1 minute data of 24 hours from 240 homes in 2 seasons. So that's a lot of data to really work with. And then we have now almost developed the set of predictor variables, which are almost 50 plus, which will be used for making this land use regression model. We have collected nutritional data, physical activity, home characteristics, SCS, indoor refrigeration source, etc. Almost 80 plus variables. Metals are quantified in hair and nails. Lung function is assessed, at least verified, validated data about 3,000 subjects. Blood, hair, nails, etc. was collected, and we were able to do that for 5,000 subjects at the beginning, and right now we have 25% of the follow-up which are done. So what I would say, if I allow you to recall what I started with. So the study, which was the New England Journal of Medicine from 2004, where they looked at the effect of PM 2.5 and NO2 on lung function growth and development in children, they stopped at 30-35 microgram per cube. We have now a range. If you don't look at the average, it can go from as low as 40 microgram per meter cube to 780, microgram per meter cube, right? So that's a very wide range which has been examined. Interestingly, unlike the other populations in the West, we have a considerable proportion of the children who are underweight, undernourished, for example, almost 2/3, right? We have now a comprehensive exposure database, which is now being exploited for developing models so that we can look at the relationship with the lung function. And we have now we were able to comprehensively examine the respiratory effects. Not examined, I mean, have the data. We are examining the effect right now, controlling for all other factors like anthropometry, nutrient intake, physical activity, SES, etc. So that's all what I wanted to share. I'll just take 1 minute for those who may be interested. So while you can see there's a massive effort here to collect this data, but the real fruits will be when this will be actually analyzed using, for example, Python, machine learning methods, both linear, nonlinear models to be built for exposure as well as health and exposure. So we are really looking for motivated and skilled and competent people to delve into some of these things. So if you have any interest in working with some of these data sets, do write to me or other investigators who are part of this study, right? And that's what I wanted to share with you, and I'm happy to take any questions, concerns, suggestions that you may have. Thank you very much. Thank you, Shiva. That's all what I have.
Harish, thank you very much. It was a wonderful talk and a very comprehensive measurement studies in India, and particularly provides and various opportunities to understand how the air pollution exposure can resulting into lungs related risk, which you really very effectively, and I'm sure that this will provide a lot of guidelines, informations to the policymakers as well as decision makers. So coming to, I just wanted to know that since there is a government of India has already have PM10 2.5 standards, and but the study shows very clearly the mass concentrations and the compositions are vary from location to the location.
Correct.
So
Correct.
hence, is there any way we should move forward, because these mass concentrations may not be reflecting, even if the mass concentration is 40 or 50% of the standard, but it is more toxic, that may cause much more danger, damage to the environment and public. So based on your expertise, can you comment on that?
Yes. So I think Shiva, I should not be telling this. I think you know it very well. I think you have been actually one of the champions to really kind of suggesting some of those based on massive amount of research you have done. But let's say from this particular study and also something which I've been doing for last 8-10 years here in Mumbai and other places around, I completely agree with you that the mass concentration, first of all, we are still regulating, even though the new regulations may come soon. As you know, we are in the process of revising our NACs. But still, I don't think we will really lower them. For example, similar to what, let's say, the EPA has done in US or the other standards. So first of all, already the levels which are being regulated, even if we meet all those standards, are not necessarily going to safeguard us. First, that is the first thing. And the second, you are absolutely right, because I think also I have really gone through some of your studies as well. See, and we have done a number of studies in tunnels and roads also. There is actually a very high proportion of dust, which is really like kind of, adding to the mass concentrations. And this dust, some of it component may be toxic, but some of them may be rather innocuous. So in that sense, I think like maybe probably we can go more towards, let's say, components like black carbon, or for example, a combination of a few metals. The only problem is that the quantification of these, I think you absolutely know it far better even than me. It is very, very, kind of resource training. I mean, the training constraint, resource constraint, and funding wise. I mean, the maintenance of these. So, but I would say that probably like what you are doing at CEPHA, and so maybe we need to use this network to kind of have some hubs, where periodic samples could go, let's say, a rather smaller frequency, and then some uh, of these partial samples could be analyzed for, let's say, black carbon, elemental carbon, or some of the heavy metals, OC, or some of those, which will probably represent a better, you can say, measure of the relative toxic component. And obviously, we'll have to then link it, right? We'll have to still see that whether those are actually able to show, for example, from hospital admissions to uh, mortality. And so I think it's um, I really am humbled when you said that it will pave way to policy. I don't think so, but yes, it may help. It may add to some additional data on that. But for definitely, it is really raising questions about the sufficiency of mass concentration, because we have already moved from PM10 to PM2.5. So what essentially we are really looking forward to is what can be better than just PM2.5 mass. And I think probably it could be a chemical component if a relatively easier method is available, which is affordable. Then I think we can really, really look forward to that.
Thank you very much. I think it's a very useful thought from your side. I just thought before we go to the next discussion, anybody from the online or any of you would like to ask questions, because then we will continue with that. Anybody would like to have any questions to Harish? Online, please type your questions in the chat box so Harish can see the questions and he'll respond to you. Any questions are there, Sharu? Okay, Anisha, the other question maybe before somebody asks questions. See that you have did extensively the chemical composition. So
Yes.
is it possible to come out with some kind of an biomarker where we can say that can we look at it as a biomarker to kind of different sources? As a result, we can say the health risk.
Yes. Yes. So Shiva, I think that's an excellent question. I mean, that is one of the intentions. So we are going to look at CC16, which is a circulatory inflammatory marker. We are going to look at some of the interleukins also. And also we are going to look at some of the heavy metals in hair and nails, etc. So, and then we'll try to relate them with the outdoor exposure of those components what we have measured. So I think it is premature right now, because even we don't have the first inflammatory marker results. We do have the metals in nails, hair, and those, but it has not been well examined right now. But yes, that may help us to see that, let's say, if certain of these are actually even the same elements, right, which we are anyway measuring in PM also, especially, let's say, metals, etc. So if these are also raised in the hair and nails, so that is, I think, going to give us some good indication. And if these inflammatory biomarkers are also raised in children who are exposed to higher levels, then they have also, so then I think that could be like a clinical marker which could be potentially also utilized. But these are some of the things which we'll have to see once the data is fully analyzed.
Very nice. So any questions from here? Anybody would like to ask some questions? Vika, Chaitra, Ruth.
Okay, it looks like everybody is very shy about it.
No, yeah. So maybe my last question from I'd like to know your comments on the lung function is something which we will also trying to see that people are for children's particularly those who are staying in a very dusty environment
Yes.
because I'm just trying to see that maybe if there is a construction activities or the locations maybe near traffic site or a industrial area. So hence they will always exposed to higher pollution levels. And will that significantly affecting the capacity of the lungs of particularly children, and as a result, their breathing response may get affected.
Correct. Yeah, I think you are absolutely right. So what you are, I think, really sensitizing us all about is that, I mean, what we are talking right now is only the environmental exposure. What you are saying is probably also occupational, or let's say certain hotspots and microenvironments where children, adults can be exposed to uh, other kind of higher levels exposure. So yes, I think like there is already a very huge literature on occupational effects. Of course all of that is on adults because obviously children more or less are not allowed to work, so there are very few studies on that. But for example, like we have been looking at in Chandrapur, near Chandrapur, there are mining areas. So one of my student, Kamlika, she has actually done pretty similar study like this. She tried to look at the effect of the haul road trucks, right, which are really transporting the material in and out of the mines. She looked at one of the major roads, which is connecting the major throughway to the city and residential places. Conducted some questionnaires. We don't have objective health assessment like what we are doing here, but we did find that the symptoms, like for example, whether they have doctor diagnosed asthma, or for example they have respiratory symptoms like cough, and I think irritation in eyes, and some of those are significantly higher in those women who were near either roads or the mining areas compared to farther away. So suggesting that wherever we have known sources of particles and air pollutants, probably people are going to expose, and it will show up in the health effects. But yeah, I think studies will be needed in let's say traffic personnel, postmen, some of those people who are really exposed to traffic air pollution all the time. I think that is definitely one interesting thing we can look at.
Harish, thank you very much. It's a wonderful work, research. Highlight these aspects of air quality in different microenvironments and their exposure. Thank you very much on behalf of CEPHA, Air Quality Management Association, and also from IISCM and IIT Madras. We thank you very much for your time and a wonderful lecture. So I request everybody to just start your camera. We will just have a have a picture with Harish, and then we'll use this in our website. Okay, so all of you just stand with Harish, and then we'll take. How do we capture it? Is it possible to capture online? Huh? Or we can do stand here, and Harish, can we make it as is it possible? How do we do that? We can stand here, no?
Stop sharing your screen, sir, please.
Harish, can you stop sharing?
Okay. So, okay, how do I do that? Stop. Okay.
Okay, now you are in the center screen. Okay, now you can take it. Okay, is this all of you just here, all of you just start your camera. take it a screenshot, or we should we plan like this only? Then it will take it better.
This camera will capture you. Yes.
Then we'll include a little bit. We'll close it then. So is that okay? So we can sit very good. That's okay.
Yeah, I think the sitting one is better, I guess. Yeah, it looks better.
Okay. Okay, you are missing. But you can open that one. One more is there. That camera open. Yes. Okay, thank you very much.
Okay. Thank you, Shiva, and everyone who attended. And Shiva, I'm looking forward to actually be there during December when you are really organizing the next, I think 8th or 9th, I guess. I don't remember exactly. Ninth conference. So yeah, I look forward to that. And yeah, those who have any queries, or also if you have interest in working with some of these data sets, do send an email to me. Thank you very much. Yeah,
Thank
bye.
you. Thank you all for joining this talk. I'll see you in the next month with some new expert. Thank you very much.
Thank you. Yeah, bye. Bye.